And so we gotta go fast. So everything about all of these, it's about energy utilization, energy, gaining energy, using that somehow, so that life can occur. Of course, all of it, whether it's photosynthesis, cell respiration, all of it does go back to enzymes and enzyme structure. So when we look at this, enzymes are going to be at least the tertiary or the quaternary level of protein structure. As those proteins fold, as they take on their particular shapes, that's going to generate several different particular structures in the enzyme. First and foremost is going to be the active site. This is going to be where the substrate binds. That is where the reaction is going to happen. That is where the substrate will be turned into the products. We also have some other parts of the enzyme. We can have allosteric sites. These can be on other areas other than the active site that things can bind to. And then that can potentially change the shape of the enzyme. And that can change it into the active form or into the inactive form. And so this is a regulatory mechanism. And we also have cofactors and coenzymes, and these are going to bind in the active site, giving it its final correct shape. So cofactors are typically metal ions. Example would be copper. You need copper in your diet, very, very, very small amounts. So you don't have to be like, I need to suck on this penny. You get enough from your food. That's necessary for the enzymes that help manufacture your red blood cells. And then a coenzyme. This is some type of vitamin. So B vitamins, which are necessary for energy production, they bind to enzymes. And so they bind, give the active site its correct shape so it can bind to the substrate so it can do its job. So when we are looking at enzyme kinetics and the way that they work, you have your active site. And so what we see in this picture, we have the original shape. But when the substrate binds to the active site, we see that the shape of the active site changes slightly. That is called the induced fit, so that there is a tighter bonding between the active site and the substrate. And then after the reaction is done, the products are released and your active site goes back to its original shape, and it can continue to catalyze reactions. So the enzyme is not consumed, it is going to be continued on. When we look at the way our reactions happen, we typically have either exothermic or endothermic. So in this, we see our exergonic or exothermic reactions. And we see that we've got the energy level of our reactants here, and our products are lower. So all of this change in energy, delta G, don't worry about what that means, doesn't matter. That is going to be released energy. So examples of exothermic or exergonic reactions in biology are going to be cell respiration and the hydrolysis of ATP. So when ATP breaks apart, that releases energy, that gives the energy to do cell work. Cell respiration is the breaking down of glucose, and that energy that is released is then trapped, runs the ETC, makes the ATP. Over here, we see that our product level is higher than our reactant level. So that means that energy is going to be stored. So that is endergonic or endothermic going in. And so our main most important endergonic reaction in biology is photosynthesis. So that's our main most important. Other ones are anytime you're building anything, if you are building a protein, if you are building starch, if you are building glycogen, you are storing energy. So those are all going to be endergonic reactions. So any kind of dehydration synthesis is going to be storing energy. Any kind of hydrolysis is going to be releasing energy. When we look at these reactions, if we were to draw in what a enzyme does, that is simply going to make the bump much lower. So what is the bump? That's the activation energy. That's the energy needed to run the reaction. So an enzyme lowers that, so our reaction happens faster and more efficiently. So it may seem that activation energy impedes biological reactions, but actually activation energy is good because that's going to help maintain a lot of molecules. One thing, so we see delta G, that's our change in energy. The reactants and your products, whether it is a uncatalyzed or a catalyzed reaction, that does not change the amount of energy in the endothermic or exothermic reaction. It just happens faster. So when glucose is combusted in cell respiration using enzymes, the change in energy from glucose to carbon dioxide and water is the same as if you would light it on fire. It's just that lighting it on fire is faster and more dramatic. So it seems like you would be giving off more energy, but you're really not. Activation energy is good because this barrier right here, that is actually going to prevent spontaneous breakdown of lots of different molecules. So we do not want our DNA to spontaneously start degrading. That would be bad. So having a high activation energy for the breakdown of DNA is a good thing. Then we need to have the proper enzymes to catalyze the proper reactions, but we don't want things randomly breaking apart when we don't want them to. So things that affect enzymatic reactions, we're going to have optimal temperature. We'll have optimal pH. We'll have an optimal salinity or optimal salt level. If you go out of this optimal range, what happens to your reaction rate? It slows down. If you go extremely far beyond this, what happens to your protein? It's going to denature. So, and then that means your protein is going to start to break down, changing its structure, and it doesn't work anymore. So when you cook, why do we cook food other than to make it taste better? Right. And we're going to denature which particular proteins do we want to denature? The bad ones, the bacteria, the parasites, whatever that's in that food that would make us sick. We want to kill that, whether you are killing it with heat or if you are killing it with acid. So if you're doing ceviche, where you cook things with lemon juice, that is denaturing that acid or denaturing those bacteria because you're getting them out of those optimal ranges and you are destroying them. Salt can do the same thing because remember that salt, when it dissolves in water, makes ions. and you're going to have those plus charges and those minus charges. And if we remember, many of our amino acids have charges. So if you add positive ions in a solution, they can start bonding to the negative charges of amino acids. And those amino acid charges are what give them their structure. And so that causes the structure to then change shape, and it breaks down the protein. So you can actually cause certain proteins to precipitate out of a solution by adding salt. We attempted to do this when we did our DNA extraction lab. We added a salt solution, the protein precipitator is what it was called. It's just salt. It binds to those extra proteins to get it away from the DNA. That part actually worked. And so the salt can affect the structure of the proteins. Other things that affect it are going to be your substrate and your enzyme concentration. So typically what happens as you add more enzyme, this is going to increase the enzyme. That's going to increase your reaction rate. Why is that?
Because there's like unlimited active sites for the substrates to find.
Exactly. So if, for some reason, we decided that we're going to pick up all the garbage around Baylor Lake, and I sent Penelope out there to do it by herself, it might take a while. But if we all went out, us being the enzymes, we'd be able to pick it all up much faster. However, if there was so much garbage, you would eventually get to the point where you are working all out, picking up all that garbage, and you are working as fast as you can, and then you can't work any faster. So as you increase substrate, you are typically going to see an increase in reaction rate until you get to the point where all the enzymes are working at full capacity. And that's known as enzyme saturation. And then even adding more substrate, you're not going to work any faster than that. So both of those can affect how quickly reactions occur. Other things that are going to affect enzymatic regulation are going to be things like competitive and non-competitive inhibition. So competitive inhibition, your competitor is binding to the active site. So that means the group that wins is the ones that has the higher concentration. So if you've got more inhibitor, the reaction slows down. If you've got more substrate, the reaction speeds up. The other type is non-competitive, where your inhibitor binds to an allosteric site. That is not the active site. And when this binds, it changes the shape so that the substrate no longer fits. So you could have tons and tons and tons of substrate and a very low concentration of inhibitor, and this will still work because they are not competing for the same spot. So if you have enough inhibitor to block all your enzymes, that could be 1,000 times less than your substrate, but you would still have an inhibited enzyme. So this is usually involved in a negative feedback loop. Where your inhibitor is the product of your reaction. So that when you have used up enough substrate and you have made enough product, you want to stop making that product and wasting energy. All right, so other things with your enzyme structure, you've got that allosteric site again. So sometimes your allosteric sites will inhibit. Again, same thing. That changes the shape of your active site, so your substrate no longer fits. But sometimes your allosteric site is going to be an activator. And what that activator does is it stabilizes the active site so that the reaction can happen more effectively. So, and this can depend on the type of metabolic pathway, whether you have both. Some enzymes will have both an inhibitor and an activator. Some only have one or the other. Yes.
Then why do we need the allosteric activation?
So what can happen is your active sites and some enzymes can fluctuate between the inactive and the active form just naturally. And then when the allosteric activator binds, it stabilizes it just in the active form, or the inhibitor will bind it in the inactive form. And so it's just kind of like, it depends. So we see both allosteric inhibitors and allosteric activators in phosphofructokinase. Which is our regulatory enzyme in glycolysis. When you've got lots and lots of ATP, that is going to downregulate your phosphofructokinase, which we're going to label PFK. But when you have low levels of ATP, that causes the production of AMP, which is then going to turn PFK on. So our ATP is our inhibitor, AMP is our activator. And so what happens if you're just sitting there and you have plenty of energy, so that's going to downregulate your phosphofructokinase, so you are not just breaking down glucose. But if you then get up and you start running, you start using up your ATP, you're going to then start manufacturing and breaking the ADP down into AMP. So it's going to kick on your phosphofructokinase to ramp up glycolysis, to ramp up cell respiration.
You said the ATP is the inhibitor?
Yes. So then again, back to cofactors and coenzymes. So cofactor coenzyme binds into your active sites, and it gives you the correct shape for the substrate to fit in. So some terms that might show up. So APO enzyme is when the cofactor coenzyme is not there, but when they bind, you get what's known as the holoenzyme, which you can also think as the whole enzyme, which makes it functional. So this is where you get into dietary deficiencies. If you're not eating your fruits and your veggies or taking your Flintstone gummies, you may not have these vitamins and minerals for your enzymes to work properly. All right, same idea. All right, so moving on to cell respiration and photosynthesis. And basically, again, equilibrium equals death, because if we do not have a constant input of energy, then the molecular work that our cells need to do cannot happen. So if you are a plant, you need that input of sunlight so that you can make enough energy to store. So at night, you can break down sugars using cell respiration to stay alive. So we need that constant input of energy. And so that's what this is showing. So this could be light energy. This could be food energy or chemical energy. And so either one of those is going to power electrons to turn our turbine, which is our ATP synthase. If you don't have those powered electrons, your turbine stops moving, you stop making ATP because your gradients are equal, and then you no longer are going to be alive. It is beneficial that we have things released in multiple steps rather than one big one, because multiple steps are going to control energy release. So we say cell respiration is 3 steps. It's not. It's way more than that. It's 3 main steps. Glycolysis is 10 steps. Krebs is that whole cycle, and then ETC, all those individual steps. If when we burn glucose, you light it on fire, it's an explosive flame, and all that energy gets released all at once, which makes it very inefficient. And so our cell processes break it down over multiple steps to harvest that energy to make it more efficient, so we get more energy per glucose molecule. Okay, so photosynthesis, the origin of photosynthesis is an ancient bacterium that is thought to be very similar to a group of bacteria called cyanobacteria. This is sometimes called blue-green algae. There's lots of different kinds of cyanobacteria. They're not all blue-green, but these can be filamentous. They can be individual cells. But basically, each cyanobacterium is a chloroplast. It's got an outer membrane, and then it's got layered membranes inside that are thylakoids. And then inside of the cytoplasm, there is rubisco, which is going to do the carbon fixation for that cell. And so then back to our endosymbiotic theory, this gets wrapped by the host cell's membrane. And so then you get your outer membrane, your inner membrane, and your thylakoid membranes. So that's the theory. And the reason that this is significant is that this is where our original oxygen probably came from these early cyanobacteria cells. And so when we look at geological evidence, so looking at these rocks right here, you can see these different layers, and then you see these rust-colored layers, and it's rust-colored because it's rust. This is iron that has mixed with oxygen. So this is when your photosynthetic bacteria became at a high enough population that they generated enough free oxygen that the iron ions in the ocean formed iron 2 oxide, which is rust, and it precipitated and formed these layers. And then it happened again. And so this is called the Great Oxidation Event, when photosynthesis became high enough to generate oxygen in the atmosphere. So your first level of rusting, and then it built up again, and then you got another level, and then now we've got enough oxygen for us heterotrophs to live. All right, so our basics for photosynthesis, we're going to photosynthesis and cell respiration in 25 minutes. You ready? All right, so our light-dependent reaction. So the main focus of what is going on here, what is important, is light energy is captured. And it is used to energize the electrons. And then these energized electrons are going to go through the ETC to make ATP. And then they are re-energized to make NADPH. The thing that is not actually important in this process is the generation of oxygen. That's a byproduct. Where does the oxygen come from? The oxygen that is coming out comes from where? Comes from the water. The CO2 that the plant is going to use, the oxygen in the CO2 and the oxygen that is here, they are not the same atoms. They are different. The there's a very, very common question on the AP test. Where does the oxygen from photosynthesis come from? It comes from water. So then over on the Calvin cycle, there's going to be all this stuff that happens in the Calvin cycle, which happens in the stroma. But the important thing is Rubisco fixes CO2 into glucose using the ATP and NADPH. Down here, this says GA3P. That is glyceraldehyde 3-phosphate. That is half of a glucose. Because we make half of a glucose, how many times does the Calvin cycle have to go? Twice. Well, there's 6 carbon dioxide, so really, so we should do a 3 right there. And then it happens twice, which gives us 6. You should also know the formula and the chemical equation for photosynthesis and cell respiration. In fact, you should know it really well for your quiz, the note content, because if you don't know that equation by now, I'm gonna rip out my eyeball.
Can you write it on there right now?
6 water plus 6 CO2 gives you C6H12O6 plus 6O2. And then going the other way is cell respiration. So this is photosynthesis. Going that way is cell respiration.
Do you think we should maybe push back this risk example?
Nope. All right, so photosynthetic pigments, your main photosynthetic pigments are going to be chlorophyll A and chlorophyll B. Looking at this chart, what colors do they absorb best?
Red.
Red and blue. This is a spectrophotometer, so this is how they figure that out. They shine light through the sample and they see which one absorbs best. You will very often see these charts on the AP test. So it is simply going to be asking like chlorophyll B absorbs what color light best, and then you like it absorbs like around 450 nanometers. What doesn't it absorb well? Anything between 500 and 625, and those are the color, and those correspond to specific colors. We also have carotenoids, phycoerythrins, phycobilins. Those are secondary or accessory pigments. And why are they important? What?
They absorb like green.
Right. They absorb other colors to give a broader antenna for your photosystems. All right, so our basic things, photosystem 1 and 2, they are going to gather your light energy. They are going to energize electrons. Those energized electrons are going to go into the electron transport chain. We're going to make a hydrogen gradient, which will then go through ATP synthase to make ATP. The electrons will be re-energized in photosystem 1. And then they will go to nicotinamide adenine dinucleophosphate reductase, and that's what makes NADPH. So that electron, so our electrons from water get energized, they pass through our electron transport chain, hydrogen gradient, ATP synthase, we make ATP. Our electrons pass to photosystem 1, we re-energize them, they pass through this protein called ferredoxin. NADP reductase takes the electrons, combines it with NADP plus and hydrogen to make our NADPH. Yes.
On our sheet, there's like a space for the thylakoid membrane. Is that?
So the thylakoid membrane, why is that important? So this is our thylakoid membrane right here.
It's where the electron transport.
It's where the electron transport chain is located, and it also, this creates your gradient. This is what separates your hydrogens from your stroma. so that you can generate a high enough concentration of hydrogen to make your ATP. All right, so why do we need the NADP H and the ATP? For what? I don't know why I do that. What are they particularly doing?
Electron transport.
We've already done that. So we've already made that. ATP is going to give the energy to bind CO2 into sugar. But in order to bind things together, what holds carbons together in a bond? What is a covalent bond? The sharing of? And then NADPH is bringing those electrons in, as well as the hydrogens. So ATP gives the energy for the Calvin cycle. NADPH gives the electrons and the hydrogens to build the sugar. And so then your end product of all of that is going to be half of a glucose. What?
What did you?
So I said, so ATP provides the energy to power the Calvin cycle. This provides the electrons. And the important enzyme is Rubisco. That is the carbon fixing enzyme. What do we mean by carbon fixing?
It's carbon dioxide and sugar.
Exactly. So carbon dioxide is a gas. It turns it into the solid of a sugar. All of this right here, this big chart, you do not need to know. The very most that would happen is they will give this picture to you on the test, and you'll have to say like, then pick information off of it, but you're not going to have to explain it. Maybe it'd be like, what's the most important thing in here? It would be Rubisco as the carbon fixing enzyme. All right, photosynthesis is kind of important. Why is it important? Just looking at this picture. Right, that is the source of all our energy. So that is for primary production. As we go from one level to the next, what's our rule? 10% rule. So only on average, 10% of the energy is transferred from one trophic level to the next. Going from sunlight to plants, however, that is only 1%. Only about 1% of the energy from the sun is captured by plants. And that 1% is all of the energy for the entire ecosystem. So when we start cutting down plants, we are limiting the ability of the ecosystem to have energy. So save the trees, people. Okay. We've got 14 minutes for cellular respiration. We are doing great. All right, so both photosynthesis and cell respiration rely on hemiosmotic phosphorylation or oxidative phosphorylation. Oxidative phosphorylation is particularly cell respiration because it's using oxygen. But both of these, all chemiosmotic phosphorylation means that we are building a gradient, energized electron through the ETC. We build a gradient, and then that goes through ATP synthase. So I would summarize those words. So in photosynthesis, our gradient is in the thylakoid space. In cell respiration, it's in the intramembranous space between the inner membrane and the outer membrane. You will not have to draw these things. Again, at most, you will see these things pictured, and you will have to kind of answer questions based on what the picture is showing you. So in cell respiration, we've got our hydrogen acceptors, our electron carriers, NADH, putting through FADH, putting its electrons through. And a very common question that happens, the final electron acceptor in cell respiration is oxygen. But then the oxygen binds with hydrogen to form water. So our final electron acceptors. Cell respiration is oxygen, which then is going to form water. And then in photosynthesis, what is the final electron acceptor? Photosynthesis, we start with water. Where do the electrons end up? NADPH. And then ultimately, they're going to end up in glucose because NADPH transfers them to the glucose. Or an organic molecule, as it sometimes puts. Yes.
What's on the other side of the internal membrane space?
What's on the other side of the inner membranous space?
Yeah.
The outer membrane.
Oh.
So, all right, so some prokaryotes can do chemiosmotic phosphorylation. However, so whether it is a photosynthetic bacterium or an aerobic bacterium, they just pump their hydrogens into the environment. Is that going to be as efficient? No, because what's going to happen to these hydrogens? If they're just being pumped into the environment, they're just going to spread out and diffuse away. So that's why an extra membrane would allow a gradient to build up and make it more successful. So that's why when the pre-mitochondrial prokaryote was absorbed in the endosymbiotic theory, that extra membrane made it more efficient, which then was a benefit for the cell that absorbed it. Okay, so glycolysis, splitting glucose. That's going to give us 2 ATP. This is anaerobic Y. What does anaerobic mean? It does not need oxygen. And this is what's known as substrate level phosphorylation because the ATP that is formed is done by enzymes. It is transferring a phosphorus from one molecule to ATP. It's not using a gradient. So when you see substrate level, so this is enzyme transfer phosphate to ATP. Aerobic respiration, Krebs, and cellular and the ETC, that is oxidative because it is going to need O2 at the end of the electron transport chain. Our hydrogen acceptors bring in the electrons, the power of the electron transport chain. Where do the electrons come from in the NADH and the FADH2? What's the source of electrons for the electron transport chain in cell respiration? Where did the NADH get it? Well, it didn't get electrons from the sun. What? It's from food, right? So when we take glucose, when we're breaking that down and we are breaking off CO2, we are extracting electrons. We are extracting those electrons that we got from water in photosynthesis, and we're using those to generate. So aerobic respiration, 34 to 36 ATP. Krebs is going to make 2. So electron transport chain is going to make 32 to 34, but I'm combining those together. Fermentation makes 0 ATP. Fermentation is important when there is no O2 to regenerate NAD+ for glycolysis. What are our 2 types of fermentation? Yep, and lactic acid. So that is to keep cells alive until oxygen can be restored. So this right here, great thing to have in your head to give you a great summary for cell respiration. Good, yes. All right. Glycolysis is going to happen in the cytoplasm. What is splitting glucose? That is because of phosphofructokinase. This is your regulatory enzyme. The products, not super important to have memorized. It'd be more of a, oh yeah, that's glycolysis. Like they're not going to say how many how many NADHs are made in glycolysis. They're not going to ask that question. They might ask it if they give you a diagram to look at. But why do we have 2-pyruvate? That's our split glucose. Glucose has 6 carbons. Each pyruvate is going to have 3. This is that substrate level phosphorylation. So these are the molecules that have phosphates on them and then transfers it to ADP. And so that's how glycolysis makes ATP. That's how Krebs makes ATP. But it's got lots of different steps. Do not need to know any of those. Just know that phosphofructokinase is the thing that regulates it. Krebs in the matrix of the mitochondria. That is breaking down your pyruvate into CO2. How? This is how. None of this you have to know. This diagram may show up in the multiple choice, and then you have to answer questions about it, but you do not have to memorize it. We are going to generate 2 ATP, and then the NADH and the FADH2. That is what's important, because that's what's going to power the ETC. That is where the electrons are being harvested from the glucose. And then those electrons are going to be given to the electron transport chain in order to make the gradient to make the ATP. Good, yes. All right, and then our electron transport chain, NADH, FADH2, entering in, and again, backing up, NADH electrons go through 1, 2, 3 transmembranal proteins. FADH2 electrons go through 2 transmembranal proteins. Why is that significant? For every NADH, we get how many ATP? 3. For every FADH2, we get 2. So 1 NADH equals 3 ATP. 1 FADH2 equals 2 ATP. Then we get our gradient up here. That's going to go through our ATP synthase. The hydrogens spin the ATP synthase turbine that gives and builds up energy, and then that puts the ATP together from ADP and phosphate. At the end, our final electron acceptor again is oxygen, and we're going to get 32 to 34 ATP. So if we don't have oxygen, everything shuts down. And then NADH and FADH2 have nowhere to give their electrons. That shuts down Krebs. That's why Krebs is considered aerobic, even though it doesn't directly use oxygen. And so then what's left over is glycolysis. But then we're going to have to regenerate NAD+ for glycolysis, and that's when fermentation kicks into gear. All right, so briefly, metabolism and body size. If you are an endotherm, which means what?
It's your body.
Right, warm-blooded. The smaller you are, the faster your body and your metabolism has to work to keep you warm. Why? Surface area volume. So you are going to lose heat faster when you are smaller. So to maintain your body temperature, your metabolism has to be faster. That also means that it is going to be less efficient, which means you have to eat more. So when you have something like a mouse or a hummingbird, both warm-blooded, both very small, they have very, very high metabolisms. And so they have to eat lots and lots of food to maintain their body temperature. Hummingbird metabolism is so fast that they actually have to go into hibernation at night when they are not able to eat food. So what we see is the relationship of body size to metabolic rate. As you get larger and larger and larger, you don't have to generate as much heat because your body keeps it inside. And so your metabolism can be a little bit slower. Questions? Yes.
Where does the discrepancy between 34 and 36?
So that goes back to when the NADH from glycolysis transfers its electrons across the mitochondrial membrane. It's random where they go. They can go to NAD plus or FAD. And if they go to NAD plus, that'll generate 6 ATP. If it goes to FADH, generates 4 ATP. So, all right, people. I will see you on Monday.